Hi, my name is Kelly Bijanki, and I'm an assistant professor in the Department of Neurosurgery at Baylor College of Medicine. I wanted to start by giving a big thank you to the organizers for putting all this together and for the invitation to speak today. I have the privilege of sharing with you a set of experiments we've undertaken to leverage the insights of stereotactic EEG in order to inform deep brain stimulation therapy for treatment-resistant depression. The first part of this talk will describe the design and philosophy of the study. The second part will discuss the intracranial monitoring period and our approach to characterization of the TRD network. And then the third part discusses our efforts at mapping the depression circuitry using multimodal fusion across imaging and electrophysiology. So just to give a brief background, there are several brain targets undergoing active investigation for the treatment of treatment-resistant depression. They're indicated here. The current study focuses on the first two, the ventral capsule ventral striatum target and the subcallosal cingulate target. We're investigating these targets simultaneously, so implanting bilateral leads in the ventral capsule ventral striatum and also bilaterally in the subcallosal cingulate. At the same time, during that surgery, we're also placing 10 stereotactic EEG electrodes, which are temporary, so they're kept in the brain for the intracranial monitoring period. And then at the end of that period, they're removed and the patient begins an open label deep brain stimulation therapeutic optimization phase. So just to say a little bit more about the stereo-EEG approach. So this approach is borrowing from a routine diagnostic practice in the surgical treatment of epilepsy, wherein temporary depth electrodes are implanted in the brain, connected to recording equipment, and then patients are monitored during a hospital stay that typically ranges from 1 to 3 weeks. Recordings are then studied to provide guidance on the neural underpinnings of the patient's disorder and to guide the next steps of treatment. The intracranial electrodes themselves are not designed to provide treatment directly. So we're using them for the same ends in treatment-resistant depression. This is the structure of the study. The study is led by 3 PIs, Samir Sheth, Nader Paradian, and Wayne Goodman. Samir and Nader are the neurosurgeons at Baylor College of Medicine in Houston and University of Texas Southwestern in Dallas. Dr. Goodman is the chair of the psychiatry department at Baylor College of Medicine. We receive psychiatric support from Sanjay Matthew and Kayla Bailey. I direct the neuroscience efforts, particularly focusing on the intracranial monitoring period at Baylor. We receive engineering support from Brown University, assessment tool support from UPMC and from the University of Chicago, and then device support from Boston Scientific. And we have also a critical collaboration with Cameron McIntyre and Angela Nager at Duke. So I'd like to show this video clip to demonstrate the process a little bit. The study enrolls patients with treatment-resistant depression that has failed to respond to a variety of prior adequate therapies. We study the underlying brain state in these cases, and then we apply initial stimulation through the deep brain stimulation leads indicated here in yellow, and characterize the change in network activity, as you can see in the recording here indicated in yellow, the change in the network that is directly evoked by the stimulation, and then also the more passive change in just the actual resting native electrophysiological state of the brain after the patient has received stimulation pulses. After 10 days of study, as you saw, the SEEG electrodes come out, the pulse generators are implanted subclavicularly, and the patient begins a chronic outpatient DBS treatment optimization phase. So in this study, we have 2 main goals. We want to understand the pathophysiological state of treatment-resistant depression. Then we want to take a data-driven approach to the development of stimulation programming parameters that are individualized to these patients from their own brain recordings. A little more information about the stereo-EEG sampling. So these electrodes are implanted broadly. We sort of designed the study that we had some brain regions that we were interested in, the dorsolateral prefrontal cortex, ventromedial prefrontal cortex, orbitofrontal, both medial and lateral, the dorsal anterior cingulate cortex, and the amygdala. This approach has evolved quite a bit through our collaboration with Dr. McIntyre at Duke. So here you can see our use in the study of the Microsoft HoloLens platform and what we call the HoloSeeg approach, which was developed by Dr. McIntyre and his lab now at Duke. So what you can see projected in the middle of the space here is the patient's actual T1 brain scan and the patient-specific probabilistic tractography, the white matter fibers of the patient's brain itself. So everyone is standing in the room together, wearing the same holographic headsets, and then we're able to see and manipulate this three-dimensional brain volume together and basically step through the surgery virtually to plan it. So we use the patient-specific tractography to define the implant targets using methods that have been existing in the literature developed by Dr. McIntyre and Dr. Mayberg at Emory. For the SCC, we are targeting the confluence of some major fiber systems, including the forceps minor, the cingulum bundle, and the uncinate fasciculus. And then we're using a similar but new approach to define the VCBS target that is based on maximizing engagement to the fibers of the anterior limb that innervate the orbitofrontal and ventromedial prefrontal cortices. And this method has just recently been published in Banks et al. 2022. And then in the holographic planning space, we're able to then use Dr. McIntyre's biophysical modeling of the volume of tissue activated to identify, for example, if we put the SCC leads in this patient in this spot, and then we turn on the stimulation, what are the fibers that are going to intersect with that volume of stimulation? Then we can project those fibers out and then in patient-specific space, plan the stereo-EEG trajectory to traverse the terminations of that fiber system that we anticipate to be modulating. And so that's the way that the final placement of the SEEG electrodes is implemented for each patient. So as a summary of the philosophy of the study, we believe that network disorders, like psychiatric disorders, demand a network-based therapy. And the use of epilepsy-style intracranial recordings provide a unique perspective into network activity and help us characterize the effects of stimulation. Individualization of imaging and electrophysiological analyses will hopefully drive improved outcomes for patients. Okay, so the next part of the talk, I'd like to focus on the 10-day intracranial study phase. So after implantation, patients are moved to the neurophysiological monitoring unit. Here you can see a sketch of how the time runs, where we start out on days 1 and 2 with pretty basic brain recordings at rest and during behavioral tasks that are related to depression, before we begin an exploration of stimulation parameter space, starting with short stimulation durations at single contact on individual leads, and slowly working up toward longer and multi-lead stimulation paradigms over on days 7 and 8, and culminating with repeats of those cognitive tasks during task-specific individualized stimulation parameter application, which lets us evaluate our capacity to differentially modulate portions of the network corresponding with different aspects of the patient's depressive phenomenology. In the next slides, I'll show some examples of our methods for decoding the neural state that corresponds with the affective neuroscience tasks that we run, and for developing targeted approaches to stimulation parameter recommendation. But first, I just wanted to spend a couple of minutes talking about the overall outcome of the case in our first patient, whose data is displayed here. As you can see, the patient underwent increasing stimulation over the period of the intracranial monitoring phase. And over the course of this, his instantaneous depression scale rating, plotted here in blue, dropped over time, with the top stimulation parameters evoking self-reported serenity, calm and focus, increased energy and social engagement. And in several conditions, the patient remarked that if he felt this way all the time, he would be much better at work and in his personal life. The patient has gone on to a phenomenal clinical response and has demonstrated a discontinuation response consistent with DBS efficacy in treating his depression. A little bit more information about our stimulation parameter space exploration. So during the intracranial monitoring phase, we apply single pulse stimulation to characterize the structure of the network. The longest stimulation that we do is actually a 16-hour-long experiment spread over multiple days in which we sample across 3 frequencies and 3 levels of pulse width, charge balanced with amplitude, and across 7 current directions. We do this individually for each of the 4 DBS leads. And when I say current directions, I'm referring to the fact that we're using the Boston Scientific Cartesia lead system, which is a segmented steerable DBS lead system. So we're able to sample from the deepest and the most superior contacts as a solid ring and a bullet tip, respectively. And then the middle 2 contacts are actually segmented such that we're able to direct current, for example, to the anterior facing stack, the medial facing stack, or the lateral facing stack. We're interested in this because it allows us greater refinement in the white matter fibers that we think we're engaging, and potentially with directionality to the orientation of the applied field that then could correspond with different engagement of different fiber systems. Then we move into a 15-second stimulation experiment, which is designed in a slightly different way with a different purpose than the 1-second stimulation. 1 second is randomized to the level of trials. So each applied 1-second stimulation parameter combination is different from the one that will follow it. This is to counterbalance against temporal drift in the recording and potential follow-on effects from one stimulation to the next. The 15-second experiment is actually designed to help us quantify behavioral effects of different stimulation parameter combinations. And so for this, we do 15 seconds of stimulation on, followed by 5 seconds of stimulation off. It starts with single lead and then builds up into a multi-lead stimulation approach. And after the application of 10 trials of these 15 seconds, like in each parameter combination, there's a brief psychiatric interview. So during the duration of this experiment, the psychiatrist is sitting with the patient, and after the period of the stimulation, the psychiatrist does a brief interview just asking, did you notice any change? How is your mood? How is your energy? Using the words that are specific to the way the patient describes their own phenomenology. And then toward the end, we do a longer stimulation. These are multi-lead for a longer duration to allow the patient to sort of get a sense of the feeling of stimulation in a more chronic way as a preview of what they might expect in their outpatient programming phase. So one of the tasks that we use in the intracranial monitoring period is called the affective bias task. It's a task in which the patients rate the intensity and valence of presented emotional facial expressions. We use this task to characterize at the level of each electrode, as shown here in these spectrograms, the response of neural activity across frequency space. And we can see here differential responses to happy versus sad facial expressions, where this on the left side indicated at the top with positive is the neural response to happy faces, and on the right panel of the spectrogram is the neural response to sad faces. We can examine then all the electrodes' responses together to see spatial patterns indicated in the right-hand column, spatial patterns across the network. And this task is showing robust patterned responses distinguishing between the viewing of negatively valenced and positively valenced facial expressions. These data and others are then used to build neural state signatures to compare against the neural responses to stimulation. We're also able to use the property of affective biasing itself in the study. So just to speak a little more about that, affective bias refers to the phenomenon wherein people interpret emotional stimuli in a manner consistent with their own internal emotional state. So patients with depression, when they're asked to rate an array of emotional faces, they'll rate them more negatively than those same patients would rate them after an effective round of antidepressant therapy. And they also rate the faces more negatively than a normative population of healthy control participants. And so we're able to use this response property of affective biasing and explore it as a function of valence and intensity of the stimuli, as well as using it as a readout of the neuromodulatory impact of stimulation on mood. Another application of the affective bias task is looking at it as a network quantification, so looking at alpha band and theta band activity on the first day that the task is implemented prior to any stimulation being applied. and then looking at it again after the accumulation of multiple days of long-form stimulation. So this is day 2 of the intraoperative period versus day 9. And so here you can see plotted in the red dots, this indicates that there's greater power in these indicated contacts for positive faces, and the blue dots indicate greater power in response to viewing negative faces. So we're interpreting this as some evidence that just the exposure to stimulation itself may be shifting the brain's response to affective stimuli sort of wholesale on the global scale. Another task that we use to characterize the neural state is the emotional brain behavior quantification, where patients view emotional video clips and rate their valence, intensity, and familiarity. Again, we're able to record across the network during the task, and we can, in the video task, we can decode the time component in panel C against the ongoing action of the video clip in panel D. So here you can see in panel D, the ratings of the ongoing action in the video clip are getting happier. Corresponding with that same time course in panel C, theta power is actually increasing in the brain network that we're monitoring. So we're able to then use these data to build a classifier, which we've been able to use to distinguish between the neural response to happy and sad video clips. And so far, these neural responses and the classifier shows good discriminability. The last part of the approach I'd like to describe is one of now several methods that we've developed for using the intracranial electrophysiology to drive individualized stimulation parameter selection. So the way the process works kind of globally is that we characterize power in each electrode and each stimulation condition. So we define a desired state, indicated in panel 1 in the yellow. This can be a naturalistically observed positive mood state. It can be the viewing of positive video clips, the viewing of happy facial expressions in the bias task, the receipt of a reward during our probabilistic reward task. And so we define anyway a desired state and characterize the power across canonical frequency bands in all of our recording electrodes during that desired state. And then we search across the neural responses in that frequency band to all of the applications of stimulation that we have tried during the stimulation parameter search. And then we're just ranking them in terms of the degree of match between matching the correlation matrices by condition and by frequency to optimize the match between the desired neural state and the neural state evoked by the stimulation parameters. And then we iteratively fit a generalized linear model to optimize the stimulation parameters to evoke that state. So this is a way of looking at the strongest contributions of the model, where we're able to see the highest beta weights, the sort of strongest contributions to the predictivity of the correlation matrix between the desired state and the stimulation evoked state. Strongest contribution is driven by electrode contact configuration, so which of the actual electrodes on the DBS lead were stimulated is the strongest predictor, and the second strongest predictor is the frequency, with smaller contributions from pulse width and amplitude. And then an exploratory approach that we're using is dimensionality reduction to try to interpret the neural state. So we're able to kind of plot and see, for example, defining a healthy versus a sick resting state, and then plotting the neural features corresponding with the different stimulations that we apply. You can see with the right SCC medial stimulation, we're not really changing the neural state very much from the sick resting state. Whereas when we stimulate the right SCC in the more lateral contact, we're evoking kind of a separate state altogether. So it's physically sort of discriminable and distinct from the well state, but it's also separate from the sick state. Okay, so now changing gears a little bit again to our multimodal investigation of mechanisms. So this is looking at methods for pairing the electrophysiology with the neuroimaging data to tell us more about the structure of the network. So this is the work of Josh Adkinson, a postdoc in Samir Sheth's lab, and Edie Solaki, a postdoc in Nader Paradian's lab. So in this experiment, we're applying single pulses of stimulation to DBS target structures. And we understand that the application of single pulses generates axonal depolarization and triggers action potentials to propagate through the affected circuitry and result in neurotransmission events in the cortex and the gray matter structures. So we understand that the single pulse stimulation is able to give rise to both orthodromic, so like along the main axis of the axon, and also antidromic, so backwards toward the cell body of any axon that is being engaged, and that it's able to drive firing through multisynaptic circuits, and that this engagement may not map completely onto the properties that are being modeled by diffusion tensor imaging. This is important because diffusion tensor imaging is being used broadly by the field for stimulation targeting, for surgical lead placement, including by our group, but then also for understanding what we think the effective stimulation and DBS is on the network. We're only through diffusion tensor imaging able to quantify the sort of monosynaptic, like structurally integrated white matter fibers that may not transmit through to the next level. Okay, so the way that we are approaching this, you can see that we can use the patient-specific probabilistic tractography to visualize the structural connectivity that's expected just from the patient's DTI. So here you can see on panel A, the volume of tissue activated engages the forceps minor and the cingulum bundle. Then when we implant an SEEG electrode in the terminations of the forceps minor, we're able to actually do the experiment, apply a single pulse on the implanted DBS lead, and then record from the SEEG lead that's in the termination area of the forceps minor. And that's what you're seeing actually then tested in panel B. You can see the stimulation artifact is very quick, and then we're seeing a positive going waveform that starts around 19 milliseconds and then a negative going phase around 40 milliseconds. So this is the evoked potential that is generated and recorded in basically orbitofrontal cortex following stimulation to the structure. And then panel C and D are showing our ways of measuring the prediction of the network from the diffusion tensor imaging, where red is indicating the number of streamlines anticipated to reach from the stimulation volume to the recording positions. And then panel D is the actual evoked potential that we were recording. So there's quite a bit of discontinuity between the two, actually, where we see much stronger responses in sort of orbitofrontal cortex and sort of a lack of correspondence to other structures that were anticipated to be structurally connected to this from the diffusion tensor imaging. And then in panels E and F, it's another way of looking at this. So across the top, the labels are indicating the listening areas, basically. So the orbitofrontal cortices, medial and lateral, the ventromedial prefrontal cortex, amygdala, anterior cingulate, and the dorsal prefrontal cortex. You can see again, sort of in the upper panel is the streamline predictions, and in the lower panel F is the actual evoked responses, where blue is an evoked response and white is the absence of an evoked response. And here you can see strong correspondence in the orbitofrontal cortices, in the ventromedial prefrontal cortex, and then a lack of correspondence actually in the anterior cingulate, where it was anticipated to have streamlined connectivity, but then we don't see the corresponding evoked potentials. This is interesting to us. So we've now replicated this across our first two patients, and we're able to dig into this phenomenon a little bit more. So just to explain this figure a little more, the labels across the top are the same sort of listening areas of the SEEG electrodes, and then the columns on the left-hand side are the different applications of stimulation. The upper is the subcallosal cingulate, and the lower is the ventrocapsule ventral striatum stimulation target. The upper half of each is the left, and the lower half is the right. And then panel A in each is the streamlines, where warmer colors indicate greater predicted streamline connectivity from the stimulation volume to the listening volume. and B is showing the actual evoked potentials from stimulation that we applied at each of the stimulation combinations shown in the index here. And so there's a lot to look at, but the gist here for what really stood out to us in looking at it is, again, this anterior cingulate thing, right, where the SCC is anticipated to generate engagement in the anterior cingulate based on the streamlines, and we see really very little of that bear itself out in panel B with the evoked potentials. It's really fairly minimal. Whereas sort of the opposite is observed with the ventral capsule ventral striatum. There's nothing in our streamline models or anything that would predict that we would have anterior cingulate engagement, and yet we actually have fairly strong ipsilateral evoked potentials, where when we stimulate the VCVS on the left, we're getting evoked potentials in the left ACC. When we stimulate the VCVS on the right, we sort of get the same thing, ACC on the right, evoked potentials. So just to recap this, so it's an interesting point of divergence between the streamlines and the evoked potentials in the dorsal anterior cingulate. Streamlines are anticipated or are able to predict robust engagement of the ACC, most likely by means of the cingulum bundle, but we see virtually no evoked potentials in that structure. Whereas the VCVS streamlines indicate there are no fiber tracts directly innervating the anterior cingulate, but we still see robust evoked potentials that arise from stimulation to the VCVS. This double dissociation has interesting implications just for our understanding of prefrontal networks that are modulated in DBS. Single pulse stimulation can give rise to action potentials, as I mentioned, in both directions. And it can also drive firing of neural assemblies through polysynaptic innervation patterns that may not just show up through the diffusion sensor imaging itself. To our knowledge, though, this study represents the first evidence of dorsal ACC being directly impacted by ventral capsule ventral striatum stimulation, perhaps providing a novel target for closed loop recording or stimulation in future trials. Overall, our findings underscore the need for a deeper understanding in both connectivity modalities, particularly focusing on reconciling the current findings with an in-depth histological study of the anterior cingulate to demonstrate responsiveness to deep brain stimulation. Going back to the bigger picture, using this multimodal integration, we're also able to sort of quantify the degree of convergence between the predictions of DTI and the actuality of the applied stimulation and corresponding evoked potentials. So we see overall a quite strong predictive relationship between the streamlines and the evoked potentials in our first two patients, here indicated as patient A and patient B. When we look at this segregated by stimulation target, we can actually see separation between the SCC and the VCVS in both patients. And now this is with all appropriate caveats that, in only two patients, looking at the receiver operating characteristics and the nature of the differences in that predictive relationship, we're not at this point able to make any claims related to the statistical significance or robustness of this, but it is certainly raising questions for us and contributing to our ongoing hypothesis testing in this area, where we're seeing a sort of a stronger predictive relationship between streamlines and the evoked potentials in the subglosal cingulate than we are seeing in the ventral capsule ventral striatum. And to step back even further, as a final plug, just in the scientific utility and validity of the stereo-EEG approach, the ongoing use of stereo-EEG in epilepsy and its use for intracranial research over the past 20 years has produced a large body of previously collected data, which then we're able to cross-compare with the new data that is coming from the clinical trial in treatment-resistant depression. So this is one of the first applications of stereo-EEG for anything other than epilepsy, but it is not the case that we have no depression sampling. So epilepsy is frequently comorbid with depression, and so because of that property and similarity in the implantation locations of the TRD study with what naturally clinically comes up in patients with epilepsy, we're able to then build our understanding and settle some really old questions about the value of intracranial recording in epilepsy patients. One of the questions was, what's the value of characterizing the intracranial electrophysiology during cognitive neuroscience tasks in patients with epilepsy when we think they have ongoing abnormalities in their electrophysiology because of their epilepsy? And so we're able to tease this apart a little bit because we can do these recordings in similar implantation coverage patients in groups with epilepsy plus depression, and patients with only depression. And we can see, for example, indications of where the network response is similar. So in response on the affective bias task to happy faces, we see a very similar response across depression, epilepsy plus depression, and just epilepsy, suggesting that the epilepsy itself is not necessarily driving the electrophysiological responses that we're measuring in this model system. So overall, the Stereo-EEG approach, we're very excited about it, not necessarily as a permanent precursor to DBS for depression, but as a data and hypothesis generating tool for the future of science, that really the data that are collected in the sample of patients in our current UH3 study are anticipated to inform our understanding of the structure of the network for many years to come. And we hope that this will advance over time into an understanding of network response to stimulation and optimization of stimulation that can result over time in less invasive approaches for patients and greater patient-specific optimization of DBS for better scientific outcomes and clinical outcomes. I just want to thank the members of my lab, Brian Metzger, the postdoc, Maddie Maki, the PhD student, research assistants Pratik Kalva and Brian Swee, who contributed tremendously to this work, and the amazing Baylor Intracranial Neuromodulation Group, led by Samir Sheth and Wayne Goodman, including postdocs and graduate students and research assistants, the neurosurgery fellows, the radiology staff, and the many, many people and grant mechanisms that have supported this work over the years. And a specific thank you to the PIs of the UH3 study, Dr. Sheth, Paradian, and Goodman, and then the key investigators, Cameron McIntyre, Jeff Cohn, Sanjay Matthew, and Dave Borton. Thanks for your time.